Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. I'm here with JP. How are you?
It's great to be here.
This weekend was an opportunity for you to speak at the Biohacker Expo in Miami. And you did a 25-minute talk, which I think we're going to do our best to kind of summarize and put together this concept that you've created, which I think is absolutely brilliant for people to understand why we age and the involvement of the autonomic nervous system, the vagus nerve being the crux of this, as potentially being in control or being out of control, leading to aging being accelerated. So today's episode, we're going to dig into the autonomic deregulation theory of aging. So I'm very excited to dig into this topic with you, kind of parse through some of the topic, little kind of tidbits that you created for the actual presentation, and share this with the world, because I think it's time for people to understand just how important the autonomic nervous system is when it comes to all of the hallmark signs of aging. So excited to dig into this with you.
I think it's going to be great. And for those people who are going to be just listening to this, we'll do our best to explain what the slides are, because of course the presentation had a series of slides that went along with it. If you're seeing it as a video, then you're going to see the slides also, and you'll hear our discussion about each slide. I'm going to try to do my best to explain what I meant by each slide first, and then we can sort of dive into the details of it. And the deck opens with a nice sort of takeoff on the Nike "Just Do It" slogan, and it's aging, just don't do it. The nice picture of somebody jogging in the background, because of course exercise is really one really great way to slow aging down. And we'll get to some of that. The next slide in the deck is a picture of a guy that some of you may recognize the name if you're into biohacking or into longevity, or maybe you're just into AI and you know the name Ray Kurzweil. Ray is an MIT grad, graduated about 20 years before I did. And he's famous now for being the Google's chief futurist. And he wrote a book, or had a quote about 20 years ago, saying that he wanted to live long enough to live forever. And that was his mission, because he felt like the AI singularity that was coming was going to be such an amazing opportunity for science to progress, that it was going to be possible to live forever. We were going to become immortal as a species. I think he was also talking about being able to upload himself into a computer. And so Ray Kurzweil is sort of a leading voice, believe it or not, in the whole aging business. And I think to the extent that he has this as his mission to live forever, he has convinced Google, with their nearly infinite resources, to put a lot of money towards trying to defeat aging. In fact, even Time magazine had a cover article, "Can Google Solve Death?" And they've got a subsidiary company called the California Life Company, which they've conveniently, or I guess sort of with a pun, called it Calico. But they're not the only ones. Other people have caught on recently, and there's a lot of tech billionaires who are dumping money, including Jeff Bezos, into a startup. Believe it or not, they put $3 billion into the Series A round of a company called Altos Labs, which is all about anti-aging. And these guys aren't stupid. These are very, very bright individuals who, while they may have more money than Greek gods, they do tend to spend it very carefully and with a lot of thought and a lot of consideration for the potential for their endeavors to be successful. They don't tend to not be successful at what they pursue. So it's really interesting that they're doing this. And you have to ask yourself, okay, what is it that has them believing that now is the time to be investing in this opportunity? And, as Jeff Bezos says, you want to fail fast. And it turns out that and we've talked about it on the podcast before, there's this discovery that was made by, and actually won the Nobel Prize, somebody named Shinya Yamanaka. And Shinya Yamanaka is a Japanese, actually he's a physician, but he's a research physician. And he did some work in 2006 and 2007, really a phenomenal amount of work. And he discovered that you could take a somatic cell, a cell like a skin cell or a kidney cell or a hair follicle, frankly, of an adult human. And by activating certain genes that were inherent in the genome, OS, K, and M, that's OCT4, SOX2, CMYC, and CLIF4, I think. So the OS, K, and M are sort of how they're abbreviated. If you activate them, they will cause the somatic cell to de-differentiate all the way back up to being almost like a fertilized egg. I mean, it's an induced pluripotent stem cell. And this was really important, and probably part of the reason why he won the Nobel Prize was he solved what was considered a pretty large controversy around the use of embryonic stem cells. Embryonic stem cells are typically harvested from aborted fetuses, and that certainly made the pro-life crowd pretty, angry, and the pro-choice crowd said, no, we need these things. And he stepped in and showed how we don't need them. We can make them ourselves. We can make them, and we don't even have to have somebody else's genes involved. We can make them ourselves and have our own genes. But the moment that he accomplished this and it became publicly known, there was a move to sort of dig in and say, well, what exactly happens when this reversion or de-differentiation, or for those math fans out there, we'll call it integration, occurs, going backwards in sort of the traditional progression of cellular differentiation. And another guy by the name of Juan Carlos Espucia Belmonte was the one who said, okay, it actually happens in two steps. There's a first step in this reprogramming is a partial reprogramming, which causes all of the epigenetic marks to be replaced in the genome back to the position they were when that cell first differentiated and became the fully differentiated state that it was in. So for example, you could take a 70-year-old skin cell and literally reverse its epigenetic drift, the epigenetic changes that occurred, all the way back to the point where it's a fully rejuvenated cell. And then, only then, after it had all of those changes occur, could you then revert it back up the chain of differentiation to being an induced pluripotent stem cell. And this is really interesting. And what Dr. Belmonte discovered was that if you only triggered the O, S, and K and didn't activate the M gene, that you could do that, and it took about 4 days. And within, if you turned them on and then turned them off after 4 days, you would have a fully rejuvenated cell. And this isn't theoretical. They're actually using this in some pretty amazing therapies for restoring blindness in people with age-related macular degeneration and other things that are pretty phenomenal. In fact, I think there's some skincare products that are coming out now, some new rejuvenating skin products that actually really do actually have that kind of effect on skin cells.
That's amazing to consider that it's as quick as 4 days that we can literally turn back time.
Yeah, and that's really, the goal ultimately is, and this is what these tech billionaires are putting their money into, is this idea that we're going to be able to find a way doesn't involve CRISPR and genetic engineering. And a lot of the work that we've been talking about up till now it's heavy duty genetic engineering. But the goal, and the next guy on the next slide is David Sinclair, another MIT guy, although he went to the dark side and went up to Harvard. He's been focusing on, can we do this with small molecules? Are there small molecule drugs, some of which the indications are that already exist? They're already being used to treat certain medical conditions that are age-related or are related to these mechanisms that we're going to talk about. So he claims that he's actually in his lab accelerating and decelerating and reversing aging in animals using both genetic tools as well as small molecules. Now, as I think he was recently on Star Talks, which is Neil deGrasse Tyson's podcast, talking about this, and one of the co-hosts said that, what we need to see is the Methuselah mouse. We need to see the mouse that, that's lived for 10 years. Of course, the technology's not been around for 10 years yet. So there hasn't been time to do that. But if he's right, if Dr. Sinclair is right, we're going to see some mice that have gone through this cycle of aging and rejuvenating multiple times, which will bode, really kind of excitingly for us as humans, that we might be able to. I think we mentioned this when we had Ryan Smith on from True Diagnostics. Might be a day when we can go in and have a 4 or 5 day battery of medications that we take, and at least initiate the process of rejuvenating our cells and our entire bodies back to an earlier existence.
Such a unique thing to think is not that far out, right? Like 20 years ago, 30 years ago, we couldn't even fathom this. And the fact that we're accelerating as quickly as we are through this. I was lucky enough to see David Sinclair speak at the A4M conference last year. And he mentioned all of this, but he talked about just only the OSK were required to create the shift backwards and to heal the optic nerve in not only mice, but he's starting to use it in humans. And so there was actual real life data and real life situations where this was practical and functional and actually working, which was really amazing. So
it is truly amazing. And it is, it's the fountain of youth. I mean, that's really honestly what we're talking about here. And I do find it kind of curious that we are seeing it sort of happen at exactly the same time that AI is having its sort of, so there really is sort of this singularity event that appears to all be on the horizon, where we have AI that's, far beyond what ChatGPT can do, which is already pretty amazing, but actually has artificial general intelligence and this fountain of youth, and hopefully, a lot of other really positive and amazing things that are on the horizon, all sort of happening simultaneously.
And isn't that ultimately the goal that Ray Kurzweil had, was to live to the singularity, right?
Well, it's funny that you said it's a great segue. Thank you. It's the next slide. This is, for those of you who are seeing it, this is what Ray Kurzweil really looks like. It's not quite the CGI image that I showed you before. And he's all in for trying to figure out how to live long enough to live forever. And if you consider the cycling of aging and rejuvenating that can sort of go on forever as the living forever piece, the first part is you got to live long enough to get there. And so the question is, how can we at least slow the aging process down now while we're waiting for these breakthroughs to take place? And, for Ray, that means, taking, supplements and other things, that are going to help him and keep him in as good a physical shape as he can be, given the fact that he's, probably in his mid 70s at this point. And what we want to do is figure out, okay, well, are those supplements the right supplements? And what is it that's driving aging? And that's, if you slide to the next slide, really talks about, what are those mechanisms of aging? And we talked about this a little bit when we had Dr. Miller on, talking about the work that they've been doing at the ITP and animal models with different drugs, looking to see what the effect is on their mouse model in terms of extending life or otherwise. He's not a big believer that the hallmarks of aging, that, there's been multiple publications. It's considered one of the major pieces of the industry or of the field. He's not convinced that that any one of these things is actually the cause of aging. But I don't think that's what they're saying it is. The hallmarks of aging are symptoms that all life forms appear, or all multicellular life forms appear to experience as a consequence of whatever aging is. That aging is associated with things like genetic mutations and epigenetic drift and dysbiosis and mitochondrial dysfunction, et cetera. And that these features of aging are things that may, if we understand them well enough, may give us a vision into what the mechanisms of aging are. So it's not that they themselves are the mechanisms, but they're sort of a pathway to figuring it out.
I think that's so key to understand, that the hallmarks of aging are not like how these are the signs, and these are the specific things that have been identified in research as being associated with the aging process, and particularly with accelerated aging, I think is really important to note.
Yeah, and to the extent that the mechanisms of aging that we ultimately determine are the mechanisms are part of this. For example, there's probably, it's not determined yet, but it's probably true that mitochondrial dysfunction and the free radicals and the reactive oxygen species that come from a dysfunctional mitochondria trying to generate ATP, but producing a lot of free radicals and reactive oxygen species, that's probably part of the overall mechanism of aging. Now, I can't guarantee that that's the case, but it's the symptom is the mitochondrial dysfunction, but it's also part of the mechanism. But, again, that's not necessarily what the mechanism is. So I thought a really good graphic, and I included in the presentation, is on the next slide, which is this. I don't even know what to call this diagram other than it's sort of a connectome of all of the different, or many of these different hallmarks, and showing how they interact with one another. They're not in isolation. It's not like back pain and a runny nose are two symptoms of the flu, but they don't really cause one another. Runny nose doesn't cause back pain, and back pain doesn't cause a runny nose. But in the case of mitochondrial dysfunction, well, mitochondrial dysfunction does lead to inflammation. It does lead to inflammation, which then is part of inflammation. It does lead to telomere attrition because reactive oxygen species attack the telomeres more readily. So this connectome starts to show you that it's a lot more complicated, the interrelationships between these hallmarks and how they affect one another. And I used the analogy of being sort of lost in the woods. You're lost. Yeah, you can see that tree and this tree, and there's different types of trees around you, but you don't really know how that forest is really constructed. You don't know if it's an old growth forest or if it's a new, if it's a tree farm. And so what we're looking for is that point or that vantage point where you can look and all of a sudden all the trees line up in this row and you realize, that must have been planted by a human being. I'm on a tree farm, that sort of thing, as opposed to, no, there's no vantage point at which this all lines up. But I think that there's some hints that these things do line up when you see this connectome. And then you hear David Sinclair talk about information theory, which is this first attempt to knit all these things together. So, his theory says that, as we were just saying, mitochondrial dysfunction drives reactive oxygen species and free radicals, that drives epigenetic drift and telomere attrition. But that those two things, epigenetic drift and telomere attrition, drive changes in the proteome that the cell is expressing and drive senescence in the cell, and then stem cell exhaustion, and all the other hallmarks start to fall out, except it's not all of them. It doesn't explain dysbiosis. It doesn't explain some of the other things, autophagy and deregulated nutrient sensing. That's not explained. So what we're looking for is one vantage point to look and say, hey, all these trees are lining up. And so what we did was we said, okay, let's go and, let's think about how we're organizing these different hallmarks, because just listing them in a wheel doesn't really give us some way of teasing it out. And so my group and I started thinking about this, and we wanted to find a better way to categorize the phenomenon. What we said was they sort of generally fall into four categories. And those categories are information, energy, and then homeostasis in sort of two subcategories. Homeostasis at the intercellular level or intracellular level, and then also at the extracellular level. And then, so the next slide sort of shows, the three of these hallmarks that fit within the energy category. Obviously, mitochondrial dysfunction is at the core of it, because all of our cells use mitochondria to generate energy. And then there's cellular senescence and stem cell exhaustion, which are sort of the consequences of this running out of energy or running out of time, running out of fuel to do what you need to do. The next category is information. I actually put epigenetic drift, even though it's not the genes themselves. That's genetic instability. That's mutations and things like that that can accumulate. I actually think that the core of it really is more about epigenetics and that drift away from a tight control over what genes are actually being expressed. And then, of course, there's telomere attrition. There's lots of interest in that, the telomeres being those protective end caps on chromosomes. So that group of three are information component or information hallmarks. The next category is extracellular homeostasis. And homeostasis in general is sort of the physics parallel would be inertia. It's your body wants to stay in sort of this state of functioning that is cyclical. It always sort of is grounded at one point or one way of doing things, but that life and the experiences we have and external forces push us out of homeostasis and we have to find a way back. Extracellularly, inflammation is really such a huge part of that. Our immune system is how we generally respond to things that are pushing us out of homeostasis. We also have hormonal communications that go between our organs and our different tissues to help to coordinate how they function together, to make the symphony of life that we as multicellular organisms exist as.
Would you tie neurotransmitters in there as well?
I do. So it's hormones and neurotransmitters. The nervous system is clearly involved, but generally, when it's talked about, it's really more the endocrine system and hormonal signaling.
Yeah, and the intercellular communication that needs to occur between cells, between tissues.
Exactly. Your adipokines from the fat cells, your leptin and ghrelin from your digestive tract, and all the rest of the digestive tract's hormones. It's from your thyroid and it's from your adrenal glands. It's all of that communication that gets dysregulated or deregulated as we age. The one that some people go, wow, it's interesting that you put that in that category is dysbiosis. But that is sort of the ultimate extracellular communication. We've got skin, we've got airways, and we've got our gut. And each one has a unique but related microbiome that is the cells that make up us interface intimately with those bacteria, viruses, fungi, et cetera, that exist on our skin, in our airways, and in our digestive tract. And how we interact with that and how we maintain that to be in a healthy state is really important because it affects inflammation. It affects our extracellular communication. It affects nutrition. It affects lots of different things. So we decided to include it here, and I think that's the right place for it. The final category, which is also part of homeostasis, is what's happening inside the cell. And I always like to think of the fact that, human beings have only really been around for a few million years, if even that, depending on where you sort of cut off when human beings showed up within the evolution of primates. But multicellular organisms go back 65 million years. Mammals go back, since even before the dinosaurs, like 300 million years. And multicellular organisms go back before the dinosaurs 500 or 600 million years. But single cells were around for billions of years before that. So all the complexity that happens inside the cell is really, really, really important. And I think David Sinclair does a really good job of talking about this at the beginning of his book, where he talks about the balance between anabolic activity, which is growth and knowing when to reproduce and to proliferate, and knowing how to manage living through nutrient deprivation and fasting and starving. And so you've got this nutrient sensing capabilities in the cell, and then you've got this ability to just sort of recycle in order to recycle things, get back to the building blocks and reuse things. And then what are you making? What proteins are you going to use those building blocks for? And all of those things can become dysregulated. So you have a loss of nutrient sensing, disabled autophagy, which is that process of recycling, and a loss of proteostasis, which is the maintenance of the right proteins being produced in the cell. You can see how that loss of proteostasis is intimately related to epigenetic drift, because as your epigenome gets dysregulated and not controlled, you start to produce proteins that aren't the right proteins. But it's not just which ones you're making, it's are they folding properly? Are they being trimmed properly? Are your messenger RNAs being post-translationally or transcriptionally modified properly? And so all of those mechanisms are part of this intracellular homeostasis that starts to get dysregulated. But as I said, as we look at these various different hallmarks, it gets really confusing how they all interact with one another. And so we got to the point where we said, we got to think outside the box here. That's sort of an expression that gets maybe overused a lot. But so sometimes you don't have to just think outside, you got to build a new one. So I love this picture. It's for those of you who can't see it, it's a picture of a goldfish bowl with a bunch of goldfish. And then one of the goldfish decides to jump out of the goldfish bowl, but right into another one. But it's a different bowl.
And the other one happens to be empty.
Yeah, the other one's empty. So he's like the pioneer. So our plan of sort of building another one was to look and say, well, what systems or what things are not being included in these hallmarks that should have been? And how do they impact, things? And the first one we went to, because we are interested in the autonomic nervous system, is the autonomic nervous system. We said,
this is our bias.
There is a control system that exists in our bodies. And we've done work. We know now that it's not just controlling your heart rate and your respiratory rate and, things like that. It's controlling things like your metabolism inside your cell. It's controlling your immune system. And we said, well, what else might it control? And if it does control it, how does being in a sympathetic state versus a parasympathetic state affect aging? How does it affect these hallmarks? Do they get better or worse? Are the mechanisms that are driving them getting better or worse in symphony with or in concert with the nervous system? And that's why that we went through each hallmark and said, okay, what's the effect that the autonomic nervous system has? And so the very first one we looked at was mitochondrial function. What happens when you're in chronic sympathetic mode, when you're in fight or flight? And the answer is, not surprisingly, everything gets worse. You have an increase in norepinephrine that leads to, and we've talked about this before, leads to an increase in reactive oxygen species being created. That damages the mitochondrial DNA and dysregulates key enzymes, things like cytochrome C oxidase get even released from the mitochondria. Interestingly, there's an enzyme called PPAR gamma coactivator 1 alpha, and it's involved in mitochondrial biogenesis. So one of the ways that your cell recovers from mitochondrial dysfunction is just making more, just reproduce the good ones, and the bad ones you can get rid of. You can degrade them. But what we end up seeing is that when you're in a chronically stressed state, you lose the ability to make new mitochondria. So you actually have an impairment of this biogenesis process. Another way that mitochondria sort of become more efficient is like little bacteria themselves, because of course they were originally independent. They can fuse together or separate and undergo fission sort of willy-nilly, but it's obviously very coordinated when things are healthy inside the cell. And what we see is that process gets deregulated and ends up becoming imbalanced, and you actually end up with energy production becoming inefficient in a chronically sympathetic state. There's even loss of calcium control. Mitochondria are really important for buffering calcium inside the cell. And mitochondria can actually end up, degrading and dying as a result of loss of calcium control. And all of that happens during chronic sympathetic activity. The exact opposite happens when you're in parasympathetic mode. So if you think about it from the standpoint of, well, when you reduce norepinephrine and the cortisol levels that are associated with fight or flight, you end up promoting the antioxidants. And we've talked about that before. We've talked about the fact that when you activate the parasympathetic, you increase melatonin production, you increase glutathione production. The enzymes that are associated with making those get upregulated. It turns out that PPAR gamma coactivator 1 alpha also gets promoted. So you're getting more biogenesis of mitochondria. You're getting a proliferation of mitochondria. So that's good. It also helps to regulate that fission and fusion process. So the quality control of mitochondria comes back into play. And of course, you see a better buffering of calcium. So across the board, at least with respect to mitochondria so far, we see that chronic sympathetic activity is worsening mitochondrial dysfunction, and parasympathetic recovery mode is helping restore it. If you go to the next slide, we looked at the immune system. And if you've been a longtime listener of our podcast, I probably don't have to go through much about this, but basically, when you're in chronic sympathetic activation mode, you are promoting inflammation. Inflammation is going to become higher. Your cytokine levels, circulating cytokine levels are going to rise. You're going to see dysregulation in the CNS, in the gut, in the spleen. You're going to see impaired wound healing. You're leaving yourself in the inflammatory phase of the inflammation process, as opposed to resolving, getting through the resolution phase, which is tissue repair and positive fibrosis. You end up with negative fibrosis, bad fibrosis, organ fibrosis when you're in chronic sympathetic activation mode. We've talked about this many times. You activate the vagus nerve, which is the parasympathetic recovery mode. You are activating the cholinergic anti-inflammatory pathways, plural. So the CNS neurotransmitter imbalance comes back into play. Your gut endothelial barrier is not so leaky. You're supporting T regulatory cells and a more positive relationship with the microbiome, which we'll get to also, and supporting all those antioxidant production. So not too much that we have to go through here because we've talked about this so many times on the pod,
And just quickly on this particular one, talking about how immune cells produce energy in whether it's glycolysis glycolysis and oxidative phosphorylation. I don't know why that was out of my mind, but when you're in chronic sympathetic mode, it's much more glycolysis. When you're in parasympathetic recovery mode, it's much more oxidative phosphorylation. So it's a cleaner fuel. There's less residue. There's less reactive oxygen species and oxidative stress that's produced as well. So just fun little add-on for that one.
Yeah, no, and that's why we see when you're inflamed, you're probably relying on, and we know immune cells that are activated, they're using glycolysis for energy. You're seeing mitochondrial dysfunction.
Yeah, and this is a topic that's being championed right now by Dr. Jeffrey Bland on the topic of immunometabolism and how when the immune system is driven in either direction, more towards that chronic sympathetic state, that the metabolism is far less efficient, far more activating of reactive oxygen species, far more promoting of inflammation, far more in the fight state, and the cell danger response is turned on. So that's a really important piece to the puzzle.
Yep, completely. He's absolutely right. The next slide is about epigenetic drift. And again, if you're a listener to the pod, you know how much I love epigenetics. I love talking about this. It's just such a cool system that evolution gave us. It's got three major parts to it. One is the methylation of the DNA directly. Generally, DNA methylation is silencing genes. We know that there's a lot of interplay between genes, and one gene may actually be producing a protein that suppresses other genes from being expressed. So if you suppress the suppressor, you're actually activating more of the other. So it's complicated, but altering DNA methylation is aging. I mean, you're deregulating your control systems, and chronic sympathetic activity does that. We've seen that because it's silencing, really important things like histones modification enzymes, things like histone acetyltransferase, and the same thing is true of DNA methyltransferases. They're being dysregulated. It also dysregulates mRNA levels. It pushes specific mRNAs, mRNA 155 and 146A, that are associated with inflammation and damage and dysregulation of protein production. You end up seeing them upregulated. And remember, we talked about PPAR gamma and PPAR gamma coactivator 1 alpha and how chronic sympathetic activity suppresses it. Well, you're also deregulating PPAR gamma itself. And that's going to affect insulin levels, which we'll get to with respect to intracellular or extracellular communication dysregulation. But across the board, chronic sympathetic activity is going to drive epigenetics and your epigenetic controls away from where they started. And so you're going to end up producing proteins that you shouldn't. You're going to end up with growth signals that can crop up that shouldn't be. And you're going to, at the same time, be silencing things like your P53 and BRCA1 tumor suppressor genes. So it can lead to cancers. And what we see when you activate parasympathetic recovery is a normalization of DNA methylation. Instead of deregulating P53 and BRCA1, you actually end up getting those things promoted, along with things like SIRT1, which is really good for mitochondria, also FOXO3, which is part of the whole immune cascade. So all of these things become normalized, and you're sort of maintaining your homeostasis. Your your information is being maintained and utilized. The gene code is being used properly. Same thing is true with histones and the microRNA expression. Instead of upregulating 155 and 146A, you're upregulating 124 and 132. For those of you who want to dig into that, we can certainly do that, but it's probably a little more than we need to get into on this podcast. But drop us a line, and happy to give you papers and stuff to look at. I think the next hallmark is another one, and I actually combined two on this slide: nutrient sensing and autophagy, because they really go hand in hand with one another. And just to briefly review, nutrient sensing that happens inside the cell happens usually through a protein complex called mTORC1. mTORC1 has at its core mTOR, which is the mechanistic target of rapamycin, which is a protein that has the ability to actually sense the levels of certain amino acids, things like leucine and methionine, and in its complex form, because it's got other proteins that bind to it, and they have sensing mechanisms also that then trigger mTOR to drive growth. And so when you want to have a healthy balance between growth and autophagy, and autophagy is the other side of this, which is when there's not nutrients present, you want to be able to recycle things that you no longer need, proteins that have either been broken down or aren't needed anymore, or genetic material that was used and now doesn't need to be used, mRNA that was used to transcribe and make proteins isn't needed anymore. You want to take all of that material, organelle pieces that are not functioning properly, and you want to degrade them back down to their building blocks and then reuse them. And it's a really wonderful way to get rid of things that are broken and cluttering up the space and allowing the cell to become more efficient and rejuvenated in a sense. Chronic sympathetic activity deregulates mTOR. You end up with either a perpetual growth signaling, which can lead to metabolic dysfunction and cancer, or you can have the opposite, which is almost like a wasting disease where you can't grow, even you can't heal. And that's deregulating a couple of other systems, one of which we'll talk about more maybe later, but it's called the ubiquitin proteasome system, or UPS. It's like what Brown can do for you. It's one of the ways in which the cell degrades proteins and other things that it doesn't need anymore outside of autophagy. On the other side of things, how do we get autophagy to start? We get that by seeing the levels of AMP. So again, the currency of energy in the cell is ATP, adenosine triphosphate. There's 3 phosphate groups on it. When you sort of give energy to a reaction, it means that the ATP is donating one of its phosphates, and as a result, it becomes ADP. ADP is then recycled in mitochondria to become ATP again, because the mitochondria are actually using fuel and energy to add that phosphate group back on. Okay, but when there's not enough fuel out there to do that, and the mitochondria are sort of running slowly, one way that the cell can make energy, which is not generally used, but it is under these circumstances, is it takes 2 ADP and brings them together and transfers one of the phosphates from an ADP to the other ADP, and you end up with, instead of 2 and 2, you end up with 3 and 1. And the 3 is an ATP, so you've generated an ATP, but at the expense of turning an ADP into an AMP. adenosine monophosphate. And when the levels of AMP start to rise, then there's an enzyme, which is AMP kinase, which recognizes this and then triggers autophagy. It says there's not enough nutrients here. We've got to go recycle in order to provide the cell with the nutrients that it needs. That gets completely out of whack because the production of the AMP kinase itself is suppressed by chronic sympathetic activity.
Is constant or overconsumption of food one of the drivers of this?
Yeah.
Where we're not giving the opportunity for AMP to be produced, right? In a very crude sense, yes.
No, I think it is absolutely true that chronic or constant accessibility to food that we have in Western society and the loss of almost this spiritual sense of fasting. I mean, all major cultures, in the past had fasting as something that was considered a rite of passage or a part of the religion or part of what you did. Ramadan is a perfect example. I mean, you fast all day and then you eat only at night. And that is a really healthy thing to do for a month.
It's essentially OMAD for 1 month, right? 1 meal a day for a month in theory, yeah.
Exactly. It's basically a religious, it's taking intermittent fasting and making it a religious rite.
And this happens through Lent. This happens through there's every religion has something here that's driving this particular thought process. And we do realize, and I think a really important thing, and I'll just be really quick here, is we run in cycles of consumption and degradation, right? Like consume, bring it in, but then recycle it, utilize it, and then eliminate the waste. And we've gotten to a point, you mentioned, in Western society where we're constantly eating, where we're meeting people for meals, where we're having 3 square meals and 2 snacks per day because that's what's being served to our kids in daycare or school. These are habits that we're forming that are not allowing that cyclic nature of feast and famine or feast and fast to happen. And I think fasting is a tool that we can really utilize to help upregulate this particular piece to nutrient sensing and autophagy.
Yeah, and we're spending a lot of time on this, which is great because it's really super important. When you eat constantly, your insulin levels never drop. And insulin is, insulin is sort of like a detoxifying thing because insulin is being produced in order to jam sugar into cells because sugar in your bloodstream is a bad thing. And frankly, sugar in cells isn't such a great thing either because sugar tends to be sticky and it sticks to proteins. That's why we measure HbA1cs. It's a measure of how sugary your blood is, how sugary your hemoglobin is. And if it gets above a certain percentage, you're diabetic. So again, the idea of being able to regulate your AMP kinase and your mTOR functions on a regular basis in a controlled and proper way is really important. And chronic sympathetic activity just screws it up. Parasympathetic recovery, I've written a couple of book chapters on the importance of this and talked about it in my book, the Vagus Immune Connection. It's really important that we have this balance between mTOR activation and AMP kinase, and vagus nerve stimulation or parasympathetic activation helps to regulate it. It's not that we don't want those two things functioning. We want them functioning in due course at their right times. Just have to have it regulated properly. So we've spent a lot of time on that. Proteostasis. This is a relatively easy one to go through because we've talked about a lot of the pieces of how we make certain that the cell is producing the right proteins. Obviously, epigenetics are involved. Inflammation can get deregulated, and nutrient sensing can certainly upregulate and downregulate protein production and leave it stuck in too much protein being produced, or disabling autophagy can lead up to a buildup of proteins that have been damaged. But one of the things that is really important is making certain that proteins fold properly. Okay, so going back to high school biology, you've got your gene, it's been transcribed into a piece of RNA. That RNA gets modified a little bit and then taken out of the nucleus to a ribosome. The ribosomes tend to cluster around the endoplasmic reticulum, and the ribosomes are used. I mean, I had this vision in my head of sort of one of those machines that you used to have to make little stickers that told you what, little labels, a label maker. Information goes in one end, and out the other end comes this strand of connected amino acids that are associated with that code. But those amino acids have to then fold up in a very special way in order to make a three-dimensional structure that has all of the right shape and all the right grooves and all the right charges on it. And that process isn't necessarily always done well. And so we have proteins, and for interesting reasons that we don't have to get into, they're called heat shock proteins, or chaperone proteins. And a few of them are really important for folding, that initial folding or refolding. There's also the unfolded protein response, which is a separate set of genes and proteins that help to unfold and refold proteins that have not done it properly. All of these mechanisms are deregulated when you're in chronic sympathetic mode. I wish I could tell you exactly why the evolutionary reason was for that. I don't have a clear answer for it, but at least I do know that that's what happens. Chronic sympathetic activity is deregulating our ability to make certain that our proteins are folding properly. And how are you going to know whether or not you're making the right amount of protein or whether or not you have the right amount of protein, and if you're not folding them properly? Interestingly, again, parasympathetic recovery mode does exactly the opposite. It upregulates the production of heat shock proteins. It enhances the unfolded protein response and endoplasmic reticulum functions, in part because of that calcium balance that we were talking about before with respect to mitochondria. Endoplasmic reticulum is exceptionally sensitive to dysregulated or deregulated calcium levels. And finally, there's that UPS system, what Brown can do for you in terms of recycling proteins. Again, parasympathetic recovery mode increases a component of that called E3 ligase, and there's many E3s, expression to help degrade proteins that are no longer needed or damaged. So across the board, chronic sympathetic activity is causing problems, enhancing these aging symptoms. Parasympathetic recovery is restoring them, reducing the symptoms or the effect of aging. The next slide is genetic stability. And there's lots to do with mutations that occur as a result of reactive oxygen species, and we've already talked about that. Reactive oxygen species come in and they can cause point mutations, they can cause double-strand breaks, they can even cross-link DNA. All of these things are damage that's sustained by DNA. But we have ways of fixing our DNA when these things happen, because there are reactive oxygen species, there are things that damage our DNA. So we have DNA damage repair systems that are designed to, for example, excise that have been altered. We have ways of going along and proofreading DNA that if there's been a double strand break, it'll repair it. Or if there's two sections of DNA that have broken off from one another, how do we bring them together and make certain that we're bringing them back together the right way? All of these mechanisms, it's amazing, across the board. All of these are deregulated when you're in chronic sympathetic activity mode. It's literally like the system just says, you are not supposed to be stressed out all the time. And if you're going to do that, we're going to break down. You get back into that parasympathetic recovery mode, all of those DNA damage repair mechanisms are enhanced, and you have antioxidants that are limiting the reactive oxygen species. So not only is the level of DNA damage being reduced, but you're also enhancing the DNA damage repair mechanisms for the limited amount of damage that is still occurring. One way in which DNA gets damaged or chromosomes gets damaged is telomere attrition, which is the next slide. When you have a lot of reactive oxygen species being created, these end caps, which tend to be more susceptible to damage because of their location and their levels of guanine that are in their repeats of lots of guanines, that damage tends to cause the telomeres to shrink. and because the DNA damage repair mechanisms are less effective at those end caps. Now, we have in our cells, especially in stem cells, this is really important for stem cells, there is an enzyme called telomerase, and telomerase can regrow your telomeres. What do you think damages and impairs telomerase expression?
I'm going to go with inflammation and sympathetic activation, chronic.
Right. And cortisol. It's impairing. It actually even, it does, it's even worse than simply not promoting the production of telomerase. It actually promotes the methylation of telomerase, which means in the long run, even after you do get out of chronic sympathetic mode, you're still stuck with that promoter sequence or that gene being sequestered. Now you can't make enough of it. So it's sort of like the gift that keeps on giving. You stay in stress for too long, and now you can't get out of it. You're stuck with that telomerase not being available. Fortunately, there are ways to reverse that, and you can increase the production of the telomerase. The gene hTERT can actually be enhanced through parasympathetic recovery, reduction of reactive oxygen species, an increase in nitric oxide production, as well as, again, we talked about SIRT1 before, but also SIRT6, really important proteins in the protection of telomeres, among other things. The next couple of slides, we probably don't have to go into too much detail. Cellular senescence is a fascinating subject. Cells that are starting to get either telomere attrition has gotten to the point where you're starting to see genes themselves not be reproduced, and you have a dysregulation in the proteasome. You're not making the right proteins. Epigenetic drift has taken its toll. You would expect a cell like that to recognize that it's damaged and commit cell suicide. What we've got instead are mitochondrial dysfunction, so that the cell suicide mechanism is disabled. The cell can't get rid of itself. And so it does a couple of things. One, and this is actually, I sort of feel bad if you can sort of anthropomorphize them. I feel bad for these cells. They know that they should be dead and they should be recycled and they should be gone, but they're not. So what our cells do is two things. One, they stop reproducing. They arrest their cell cycle. So these senescent cells, that we call them zombie cells, they can't reproduce, they can't function very well, but they're just existing. They're almost like pre-necrotic cells. And what they do is they start to produce inflammatory cytokines. They spit out inflammatory markers, which you would think would be a signal to the immune system to come along and clear them away. But again, in chronic sympathetic activation mode, even though you have a high level of inflammation, that inflammation isn't clearing these cells out, and the immune system isn't clearing this out. And this is worth digging into just for a second. One of the functions of the immune system is to clear out cells that need to go away. They do that in a non-inflammatory state. It's called efferocytosis. Okay? Efferocytosis is disabled when the immune system moves into that inflammatory state. So chronic inflammation leads to more cellular senescence because those immune cells, those tissue-resident macrophages, are supposed to be clearing away these old cells, but they're not because they're inflamed. What does parasympathetic activation do? It lowers the level of inflammation. It allows those macrophages to do their job, clear out these senescent cells. It also restores, to some extent, mitochondrial function in these senescent cells so that they can commit suicide. They can go through that process of apoptosis that you want them to. So across the board, again, chronic sympathetic activation, for all of these reasons we just said, makes cellular senescence more of a problem, and parasympathetic recovery clears out senescent cells. Stem cell exhaustion is again a big problem. We've talked about it with telomere attrition. You are leading to a suppression of cell reproduction, and stem cells their whole function is to reproduce and to create new differentiated cells. And so what you end up with is when you're in chronic sympathetic activation mode, you have an imbalance in knowing when to grow. We talked about that with nutrient sensing and autophagy. You're getting to some degree of cellular senescence because of telomere attrition and an arresting of the cell cycle, so they can't do it anymore. And just the general high metabolic rate that you're seeing inside these cells just deregulates a whole bunch of different cell renewal signaling you need in order to generate more stem cells or generate more differentiated cells from the stem cells. Chronic sympathetic activity is making all of this worse. Parasympathetic recovery mode starts to, through the release of acetylcholine, upregulates some really important genes, FOXO3, FOXO4, it doesn't upregulate proliferation. It allows the cells to know when to proliferate properly. So across the board, I don't want to delve into this for too long. Across the board, parasympathetic recovery is helping with this. We've talked about the effect of sympathetic activity on metabolism a lot. And since metabolism is a really big piece of extracellular communication, don't necessarily want to go into a deep dive into the thyroid and leptin and ghrelin signaling. But I do want to spend a little bit of time talking about something that I don't think, get enough airtime when we talk about aging, which is the extracellular matrix. So, we think of our bodies as made up of cells, but there's a lot of other material in which those cells reside. And there's an extracellular matrix. And whether or not that's robust and healthy, collagen fibers, et cetera, or it's broken down and damaged is a function of, we see it in our own skin and or organ health, et cetera. You want to have a robust extracellular matrix. There's a protein or a class of proteins called matrix metalloproteinases or MMPs, and MMP9 is a big one. These start to break down the extracellular matrix. Now, you need that sometimes, especially during wound healing or the creation of new blood vessels or things like that, angiogenesis or neovascularization. But MMP expression in a deregulated or dysregulated fashion can lead to the breakdown of this extracellular matrix and an acceleration of, wrinkles and damage to organs, et cetera. Chronic sympathetic activity upregulates MMP levels. Again, wish I knew why. Maybe it's in preparation for tissue damage. Maybe it's a fight or flight response. It's expecting actual physical damage. But when it's not there, it's going to attack the healthy collagen and elastin and other things that are holding our cells together. You see it in the gut with leaky gut syndrome. There's a breakdown of the extracellular glue that's holding the cells together. Fortunately, parasympathetic recovery upregulates the production or the health of the ECM by suppressing the production of these matrix metalloproteinases. You're protecting the collagen and the elastin and the other connectins and other things. It also increases hyaluronic acid and proteoglycans, which are part of that extracellular matrix. So you're not only protecting what you've got, you're making more of it. So really, same message, chronic stress, fight or flight. If you stay in there for too long, you're damaging your extracellular matrix, you're damaging your extracellular communication systems, and you're going to end up with accelerated aging.
Just to quickly add to that, this particular slide actually explains why Ehlers-Danlos syndrome and where there's challenges with collagen and elastin and the functionality of that extracellular matrix being deregulated or genetically prone or primed to be in this breakdown state where there is some sympathetic activation or where the vagus nerve is less capable of sending its signals. We have more of these issues. So that really explains that triad of POTS, Ehlers-Danlos, and mast cell activation, because we're really driven towards that sympathetic state altogether.
And I'm going to riff off what you just said even more, because I at the expo, I had the opportunity to talk to somebody. We were talking about Ehlers-Danlos, and then we were talking about a connective tissue disorder that is similar to Ehlers-Danlos, but is something that is acquired. Ehlers-Danlos is a genetic condition. The actual genes for making collagen are mutated. But they're talking about this condition in which the symptoms are very similar, but it's an onset typically after some major trauma or emotional or bacterial infection or some other event that triggers you, much the same way we see with things like fibromyalgia and what I call multisymptomatic syndrome, where these patients have all the symptoms of like the flu or fibromyalgia that all came on as a result of a traumatic event. One of those things is a deregulation of collagen production and a dysregulation of the extracellular matrix. And here's the piece that I just, you hear it and you go, oh my God, it's like a light goes off. At least it went off in my head. Collagen is deposited and built based on a gradient of your autonomic nervous system. So there's a gradient of norepinephrine and acetylcholine, and collagen fibers grow. And you could imagine they're not going to grow properly. You're going to end up with a connective tissue problem that isn't because of a genetic problem. It's because of a chronic sympathetic activity problem. And you're going to end up with a similar Ehlers-Danlos-like syndrome that exists because your autonomic nervous system is dysfunctional.
Connecting a lot of dots here. This is huge.
I mean, just like the light goes on, and you're like, yep, all those trees are lining up down this road. I see them. We're in a tree farm. I get it. Let's jump to the next slide, which is the last of the 12 hallmarks, which is dysbiosis. And I saved it for last because so many people think of it as a separate thing, but it's so interesting how your immune system, how your metabolism, how your insulin levels, how your mucus production, all of these things are affected by your autonomic nervous system. And it doesn't just affect you, it affects your microbiome. So things like which type of bacteria will exist and survive and function best in your gut is a function of things like pH. It's a function of like, what level of mucus production you're making, what level of bile there is in the digestive tract. I mean, it reacts to things like your inflammatory markers and cortisol levels. All of these things, it doesn't just affect what species are there. It can also even affect how they're functioning. So for example, cortisol can affect fiber fermentation, which we need to make, unless you're getting enough short chain fatty acids in your diet through a carnivore diet, you're going to need to make some of those short chain fatty acids from fiber fermentation. But stress prevents it. So you're literally, we probably all know this, but stress can change how your digestive system works. You might not digest things as well. Well, one of the reasons you're not digesting them as well is because you're supposed to be fermenting some of that fiber and you don't. So it passes through you. I don't want to get too into the gory details of that, but you can imagine what I'm talking about here, that you're literally not digesting things fully as a result of being in a chronically stressed, sympathetically activated state. Again, parasympathetic recovery is, first off, it's enhancing peristalsis. So you're enhancing the speed with which things are moving through your gut, because serotonin, 90% of the serotonin is created in gut. Serotonin was originally discovered as part of the process of moving things through the gut as a neurotransmitter for the enteric nervous system before it was ever found in the brain. And parasympathetic recovery is going to upregulate liver function, so you're producing the right amount of bile, digestive enzymes in the pancreas, the mucus, that's produced on the side walls of the gut. All of that is being re-regulated by the sympathetic nervous system. We call it the brain-gut axis for a reason. I mean, the ability of parasympathetic activity to not just move things through the gut, but control how the gut functions. We talked earlier about the epithelial layer and maintaining those tight junctions through support of the extracellular matrix. Well, that's really important as well. I mean, that's part of our interaction with the microbiome through the immune system. All of that regulated by the autonomic nervous system. Fascinating stuff. So this next slide, and I wish everybody could see this slide. When this paper comes out, this is the one slide to look at. This is basically showing the 4 quadrants of the hallmarks: energy, information, and the 2 in homeostasis, and showing how the autonomic nervous system sits at the center of this. And there's arrows going out in all directions, showing the worsening of these or acceleration of these aging symptoms by sympathetic activation and the deceleration or reversal of them through parasympathetic activation. And so my message to Ray Kurzweil, my fellow MIT grad, here's my message. You're taking all those pills. Great. Glad you're doing it. Hope you live to be 125 before you need it, reprogramming. But don't forget about your parasympathetic nervous system and getting into parasympathetic recovery mode. Because if you don't do that, you're not really going to benefit from all those supplements you're taking because you're just accelerating the aging instead. And so, we've talked about this before, but the last sort of the slides are, how can you do this? How can you increase parasympathetic activity? Well, obviously diet's really important. We talked about on the next slide, we talk about the polyphenol-rich diet, probiotics and fiber-rich diet for your microbiome. And we talked extensively, even on this podcast, about intermittent fasting. And I'm going to throw in the plug for ketogenic metabolism, low sugar, low carbohydrates, minimize the damage that you're doing to the mitochondria by overwhelming them and lowering those insulin levels. The next, this is basic stuff. It's diet, exercise, sleep. These are big things. And with exercise, you want to do not just, remember that exercise isn't just your body, okay? So yes, endurance training and resistance training are really important. But remember that your mind, it's where the autonomic nervous system starts, okay? You want to make certain that you're being mindful. And even more important, you've got neuroplasticity, you got flexibility in your nervous system. So do some brain exercises along with mindfulness for your mental capacity as well. It'll help with your ability to lower stress. Sleep is critical. We could do and have done episodes on this, so we won't go into too deep on this. The final is just direct vagus nerve activation. And we've known for thousands of years that deep breathing techniques, cold exposure, and meditation are great for activating the vagus nerve. The good news is we've got this device that's Truvaga. I talked about this at the expo. The Truvaga is just a great way to do it. It's sort of the modern way. I call it 2 hours of meditation in 2 minutes. You can really activate the parasympathetic for an extended period of time with just 2 minutes of stimulation. Do it in the morning, do it in the evening. It's a great way to go. And then, of course, I gave the plug out and the shout out for our podcast. But you're already a listener, so that's wonderful.
Exactly. And I think this was awesome. We got to go through all 12 hallmarks of aging. We got to go through the entirety of what the autonomic control or autonomic deregulation could occur here and the effects of that if we are chronically sitting in sympathetic state as an overarching, very simplified, hyper-simplified explanation for all of this is chronic sympathetic activation accelerates the aging process, and being able to shift back and stay in parasympathetic, vaguely activated state more often than not, stay in that state 70, 80% of the time, allows our bodies to slow the aging process more effectively by turning on all of these cellular mechanisms, cellular, intracellular energy mechanisms. It's so important to note that what we've done here, JP, what you and your team have put together here literally is an explanation for how each one of these hallmarks of aging works. I think we're really at the forefront of something huge here and how we can regulate, re-regulate, and improve the aging process. Such an important and unique topic, and just goes to show that I think we're absolutely on the right track with everything autonomic related.
I think we're getting to that singularity point. I do think that, I'm going to make light of something that's probably a very serious topic, but, in the United States right now, we're sort of looking through the Social Security rolls and finding that we've got millions of people who are over 120 years old. Well, they're not alive today. Come back in 50 years. 50 years, I bet you're going to find there are millions of people who are over 120. So
I think that's really cool to think.
Keep those bins alive. We're going to be putting people in them.
Love it. If you got to this point in the podcast, please share this episode with one person who you think could utilize this information to slow down their aging, to understand the aging process. If you're a clinician, share this with a patient who's really interested in the anti-aging process. And if you are a biohacker, somebody who's interested in living as long as possible, take this information and utilize it so that you can upgrade your health. We'll catch you all in the next one.